Whenever you are right or whenever you are correct, you feel happy and competent. But we learn something when we face something difficult. That applies here too. That applies in psychology as well. Whenever you don't know the answer of any question, then you learn more. You do mistake at the same time, but learning is more joyful and exciting, so never afraid to do mistakes. Let's begin with the session. If you're having any doubt in between, if you want to say anything in between, you can mention in chat box or in comment box. So we are having Nikki with us. Hello Nikki, hello Riggingwala. If you're having anything to mention during the session, you can mention in chat box. I will try to take your doubts, right? Let's begin. Let's move. Here you can see first question. This is question number 21st because we have already taken 2 sessions. So this is the 3rd session. So I start from question number 21st. Here you can see the ventral tegmental area primarily projects dopaminergic neuron to which of the following regions in the mesolimbic pathway. Whenever we are talking about this pathway, so there are many pathways that dopamine use and mesolimbic pathway is one of them. So question is, when any specific stimulation will be there in VTA, in short we can call them VTA, ventral tegmental area, primarily projects dopaminergic neuron to which of the following region in the mesolimbic pathway? Your options are hippocampus, external globus pallidus, nucleus accumbens and D thalamus. Correct answer is nucleus accumbens. Now the question is why? Why nucleus accumbens is the right answer? The ventral tegmental area, in short VTA, is a group of neurons located in the midbrain. You all will be aware where is midbrain is located. So when we talking about the brainstem, the upper part of that brainstem is midbrain, and it play central role in the mesolimbic pathway, mesolimbic dopamine pathway. So that is largely involved in reward, motivation and reinforcement learning. In this pathway, when we talk about mesolimbic pathway, the VTA send dopaminergic projection. Dopaminergic projection means dopamine releasing. Dopaminergic, this term means those neurons who are containing dopamine. So when we are talking about mesolimbic pathway, VTA send dopaminergic projection primarily to the nucleus accumbens, and that is the key structure in the brain for reward circuit. So whenever we feel reward, whenever we feel rewarding, at that time our VTA sending those particular information to nucleus accumbens, right? So this connection VTA to nucleus accumbens is very crucial for pleasure and reinforcement of our behavior. So correct answer will be nucleus accumbens. Next, here you can see question number 22. Korsakoff syndrome, which is commonly associated with long-term alcohol abuse and thiamine deficiency, primarily causes damage to which of the following structures within the limbic system? And your options are parahippocampal gyrus, mammillary bodies, amygdala and septal nuclei. If you know the answer, you can mention in chat box, and I will explain them, no doubt, but I want to see if you are having any doubt. If you don't know, then you can also mention your queries, your doubts. If you know, that's great, and as I always mentioning, if you don't know, that means we are learning. So if you know, you mention them in chat box. If you don't know, that means we are learning, right? So we are having these options: parahippocampal gyrus, mammillary bodies, C amygdala and D septal nuclei, and correct answer will be B mammillary body. Correct answer will be B mammillary bodies. Now we have to understand why that will be the right answer. So whenever we are talking about Korsakoff syndrome, that is a chronic neuropsychiatric condition that typically arises from prolonged thiamine deficiency, and this kind of problem we often see in those individuals who are having chronic alcoholism condition. So if any individual consuming alcohol too much, and if there is chronic problem of alcohol addiction, in those particular situations we can find that there will be prolonged thiamine deficiency. So now you are aware now why that will be the right answer. So one of the important characteristics and consistent feature of the condition is degeneration of mammillary bodies. So when any individual will develop this kind of prolonged thiamine deficiency due to chronic alcoholism, then there will be degeneration of a specific part of the brain. We call them mammillary body. That mammillary body is the part of limbic system and closely linked to hippocampus via phrenics. So that particular area that we are discussing, mammillary body, that will be the part of a specific region. We call them limbic system, and they are linked to the hippocampus via phrenics. These structure, whenever we are talking about this specific structure, this mammillary body, that play key role in memory consolidation. So whenever there will be damages to the mammillary bodies, there will be disturbance of normal functioning of Papage circuit. Papage circuit. What will happen? Papage circuit. So whenever there will be the problem in mammillary bodies, then you can clearly identify that there are disturbance of a normal functioning of this Papage circuit. Right? This Papage circuit, Papage, P-E-Z, Papage circuit is essential for converting short-term memory, STM, into long-term memory. So as a result, individual with Korsakoff is often experience anterograde amnesia along with confabutation and disorientation. So what I mean to mention, whenever there will be problem in mammillary bodies, then there will be problem in whole Papage circuit. And if there are Papage related problem, Papage circuit related problem, then individual will have problem related to memory, more specifically anterograde amnesia, confabulation and disorientation. Now the question is, I use the term Papage circuit multiple times, but what it mean? And which organ will be the part of Papage circuit? So Papage circuit is presented by one of the popular neurologists. Their name was James Papage, J-A-M-E-S, James Papage, in 1937, right? So James Papage identified a circuit. That means circuit simply means there are so many organs who are working together. So if there will be changes in one organ, they will impact so many organs, and they identified this in 1937. That's why we call them Papage circuit, because that's found by James Papage. And whenever we are talking about Papage circuit, so there are many organs who are working together, like cingulate cortex. What is the first organ of Papage circuit? Cingulate cortex. Cingulate cortex. Cingulate cortex. What will be the second? That will be ethorhinal cortex. Ethorhinal, sometimes we call them ethorhinal. Ethorhinal cortex. Along with these, there are parahippocampal gyrus, parahippocampal gyrus, parahippocampal gyrus, parahippocampal gyrus. Along with parahippocampal gyrus, hippocampus, hypothalamus and thalamus. Hippocampus, hypothalamus, thalamus. I am writing here. Hypothalamus, hippocampus and thalamus. Hippocampus, as well as thalamus, So what I said, whenever there will be problem in mammillary bodies, so there will be so many issues, right? There will be so many conditions, and whole Papage circuit will not work effectively. right? And Papage circuit, cingulate cortex, etherinal cortex, parahippocampal gyrus, hippocampus, hypothalamus and thalamus work will be impacted. And usually we are finding that if any individual having problem related to Papage circuit, then they can find problem related to the memory, more specifically anterograde amnesia, confabulation and disorientation. All these kind of problems will be there. So right answer will be mammillary bodies. Question number 23, but before moving ahead, I would like to see who are in this live. Hello Arju, how are you? Yes, I'm fine. Hope you are also doing good and enjoying your M.Phil in Clinical Psychology. Hello Noha, Pallavi, PSD Psycho, great name. Sanjukta Roy, Nikki, Noha, Survi, Chaitanya, Del, then Banisha. Hello everyone. Hello Aditi, nice to see you here. Shivani, hello Shivani Pradhan. Okay, so you all are responding. I hope, right, you are getting some new information. So don't be afraid. As I started from this particular point in the beginning, if you don't know something, then you are learning something. So if you know something, yes, you know, that means you are doing great work, you are studying, you are reading the content. And if suppose that if you are not knowing something, that is the matter of learning. That is the time when you will learn so many things. So whenever you are coming here on live, whenever you are joining my session, so I always motivate individual to comment their response, mention what do you think, which option will be correct, because whenever you are doing mistake, so you will be more attentive, you become more attentive because you did mistake, and then chances are very high that your attentiveness will help you understand that particular concept and hold those concepts for the longer period of time. So it's great to see your response in chat box. Now question number 23, you can see which of the following is not a function of oligodendrocyte. Whenever there will be a statement, multiple time many students doing mistake because they are not emphasizing on the point not. So whenever you are reading the statement, read that statement clearly, just because sometimes we are reading them but we are not processing them seriously. So sometimes we are answering like which of the following is a function of oligodendrocytes, but we have to answer which one is not. So your options are creating blood-brain barrier, B, speeding up neural conduction, C, structural support, and D, electrical insulation. So first of all, we have to understand what is oligodendrocyte. So there are two specific cells, oligodendrocytes and Savon cell. Oligodendrocytes is important, and oligodendrocytes is responsible to do myelination on those neurons who are in central nervous system, and Savon cell is responsible to myelinate those neurons who are in peripheral nervous system. Right? So oligodendrocytes are a type of glial cell, we can say, that usually you can find they are responsible for this. That means we find them in central nervous system and they are responsible for myelinating the neuron when needed. Right? When axon size will be lengthy, in those situations our axon need myelination. Right? That's why you can find myelination is necessary for sensory neuron. Myelination is necessary for sensory neuron, motor neuron. Most of the time we can find that interneuron doesn't having myelination. So oligodendrocyte is responsible. So oligodendrocytes, what I'm mentioning, oligodendrocyte is responsible for myelination in central nervous system, and Savon cell is responsible for myelination in peripheral nervous system. Whenever we are talking about this myelin sheath and myelination, so they are responsible for speed up neural conduction by allowing saltatory conduction. So first point is they help our neuron to speed up neural conduction. So here you can see speed up neural conduction will be the function of oligodendrocytes na, just because oligodendrocyte creating myelination on neuron. So when there will be myelination, their speed will go up. That means this is the right statement. So this couldn't be, but yes, this is the function of oligodendrocyte. What is the second function? They provide electrical insulation to axon. We all are aware insulation, whenever we are doing insulation on any wire, so leakage will reduce and electricity will not be enter from that particular part. So structural support is also the function, and electrical insulation is also the right answer. So speeding up neural conduction, electrical insulation, as well as they offer a structural support to neuron and help maintain CNS integrity. So we can say structural support is the function of oligodendrocyte, speeding up neural conduction is the function of oligodendrocyte, electrical insulation is the function of oligodendrocyte, but blood-brain barrier is not the function of oligodendrocytes. If you are aware about blood-brain barrier, so you are understanding what it means, and this will be created by astrocytes. Astrocytes is the responsible mechanism that creating blood-brain barrier. So we can say astrocyte help forming a protective barrier around blood vessels in the brain, and this specific barrier control what can pass from blood into the brain. That's why whenever we are consuming something, in many cases, if any individual consuming medicine as well, so that particular medicine is not passing to the brain, so a specific medicine individual needed जिससे brain barrier accept कर पाए उस component को. So what I mean to mention here, speeding up neural conduction, structural support, electrical insulation is the function of oligodendrocytes, creating blood-brain barrier is the function of astrocytes. That means this will be the right answer. This will be the right answer because we have to answer which is not the function of oligodendrocytes. So creating blood-brain barrier is not the function of oligodendrocytes. So we can say A will be right answer. Now I am moving ahead. Question number 24. In a multiple regression model, if two predictor variables are highly correlated, it may result in. So when this condition will be there, then what will happen? And we are having these options. Option A mentioning increased variance explained. Option B mentioning multicollinearity. Option C type 1 error. And option D homoscedasticity. Now we have to understand, right, what will happen, right, if this condition will be there in multiple regression model. So in multiple regression model, if two or more predictor means what we can say independent. So in multiple regression model, if two or more predictor variables or two or more independent variables are highly correlated with each other, in this particular case, it can cause a problem known as multicollinearity. What I mean to say, I am repeating it, in multiple regression model, if two or more than two independent variables are highly correlated with each other, in this particular situation, one problem will occur and that is known as multicollinearity. So you can see here our correct answer is B, multicollinearity. What is the primary purpose of using Varimax rotation in factor analysis? What is the primary purpose of using Varimax rotation in factor analysis? So here we have two keywords, Varimax rotation. We have to understand what is Varimax rotation and what is factor analysis. Then we can answer this question. And your options are A. To increase the degree of multicollinearity among variables, making it harder to distinguish independent effects. Okay. Option B mentioning to maximize the clarity and interpretability of factor loadings by simplifying the factor structure. Okay. Option C mentioning to introduce randomness into the loading distribution, making patterns less structured and more uniform. And option D to reduce the overall sample size needed for accurate factor extraction and statistical significance. One thing I would like to mention here before moving ahead, whenever you are finding any question more difficult, right, बिल्कुल भी घबराना नहीं है, right, बिल्कुल भी right ये नहीं सोचना क्योंकि this kind of question I am not able to answer, so I will not be able to answer in the future. No, that is not the case. Right now, if you are having the problem, right, just identify those problems. If you identified, okay, I am finding problems in this kind of questions, or I am having problem in this kind of concept, that means 50% race you have completed. Now, if you are having problem in current situation, that doesn't mean you will face that kind of problem, ever in the future. Work on them. Ask to your mentor, right? If you are having any difficulty, so, talk to your mentor again and again. No matter how many times you have to ask, you have to discuss to them, do so. Right? Because our mentor also feel happy when any student, when any individual doing effort to understand them, understand any question, understand any right principle and theories. So they feel happy to help right in those particular situations. So whenever you are finding any question difficult, so yes, right now you are having difficulties, problems in that particular concept, but you can handle them well, and you can understand by doing efforts in multiple way. So you can command over that particular topic, right, and you can answer them in future. So don't think in this way कि I am having problem in this particular questions, I am finding this kind of question is difficult, तो future में भी ऐसा ही रहेगा. No, right now you are finding them difficult, but on the basis of practice, information, knowledge, you can make them easy, right? So now here you can see we have to understand 2 points. Varimax rotation in factor analysis. Question is, what is the primary purpose of using Varimax rotation in factor analysis? And you are having these options. In factor analysis, what we do? What is factor analysis? We try to group related variables together into factors, right, that represent similar patterns. I am repeating it, what I mean to say. In factor analysis, we try to group related variables. If you are having multiple variables, so we are trying to group all those related variables into factor that is representing, that is the representation of similar patterns. So if there are many variables, you just combine all those variables and creating one factor, right, just because there are similarities. And when we are talking about Varimax rotation, so Varimax rotation is a technique that we use after extracting those factors. So we extracted the factors, that means there are multiple variables, and we identify multiple variables are similar to each other. So we are creating factors and we are putting all those variables in that particular factor because there are similarities. So this is the use of factor analysis. And when we talk about Varimax rotation is a technique that we use after extracting those factors, and the main goal of Varimax rotation to make output easier to understand. So kabhi bhi agar in the future if you have to think about Varimax rotation, so Varimax rotation we are using after extracting factors, and the main goal of Varimax rotation is to make output easier to understand. So these two things you have to keep in mind. Now I'm just giving you one example. So by those examples you will be able to understand what is the right answer. Now when you will read this, after this example you will be able to identify and understand what is the answer and why that is the answer. Imagine now we are understanding what is factor analysis and what is Varimax rotation in brief. Now imagine you are studying students' behavior and you are collecting data related to 6 different things. What I mean to mention, you are studying students' behavior and you are going to collect 6 different kind of data. Number 1, stay focused in the class. That means child focus level in the class. So first point is stay focused in the class. Second is complete homework on time. They are completing or not completing. So stay focused in the class is the first point that we are studying. Complete homework on time. This is the second that we are studying. Third, studies regularly or not. So studies regularly, we are taking this and collecting data. Feels anxiety before the exams, we are collecting data about it. Worries about result when they participated in exam, after exam, are they worried about the result? We are collecting data related to it. And sleep is poor during exam or not. I'm just repeating it. We are having 6 factors, or we can say 6 things that we are studying in students. Stay focused in class, complete homework on time, studies regularly, feels anxiety before exam, worries about result, and sleep is poor during exam. So these are the variables, these are things we are studying, all these. Now whenever we are using factor analysis and Varimax rotation, what we will do, we will convert all these variables, these into factors. 2 factor हम choose करेंगे. One factor will be study habits and another factor will be exam anxiety. So there was 6. Stay focused in class will be part of study habit. Complete homework on time will be the part of study habit. Studies regularly will be the part of study habits, but feel anxiety before exam will be part of exam anxiety. Worries about result will be the part of exam anxiety. And sleep poorly during exam is the part of exam anxiety. So what we did, we studied 6 things, but we grouped them into these factors, 2 factors, study habits and exam anxiety. And whenever we are using it, we can say that is factor analysis, that is the function of factor analysis. And whenever we are doing it, we are making our data more interpretable, more easy to understand. So we can say after extracting all those factors, we are using Varimax rotation to make them easy to understand. Now whenever you will read all these options that we are having, and whenever you are reaching at B option, so here you can see to maximize the clarity and interpretability of factor loading by simplifying the factor structure. So what we do through Varimax rotation, as I already told you, we are using, and main goal of Varimax rotation is to make the output easier to understand. So whenever you are moving with all these options, you can find the B will be more suitable. B is the right answer, right? Okay, so Dell mentioning, sir, is this Varimax rotation in factor analysis from statistics or personality chapter? So this is from statistics. But whenever we are talking about statistics, so no matter you are doing a study related to personality, related to abnormal psychology, or related to human development, we can use all these techniques over there. Fine. Okay. So mainly we can say that is the part of statistics. Hello Aisha. Hello Alia. Okay, so you all are responding. Great to see you. Okay, so now we can move ahead. Question is, a chi-square test is most appropriate for which kind of data? Chi-square, we all are aware what chi-square is. Chi-square is a hypothesis testing, and their symbol will be x square. This is the symbol of chi-square. So question is, a chi-square test is most appropriate for which kind of data? If you want to find hypothesis, right? In those particular condition, you are having, so many technique to find hypothesis, to understand null hypothesis. Question is, chi-square test is most appropriate for which kind of data, when you will use, and in which situation chi-square will be more suitable. And your options are A, continuous and normally distributed, B, nominal categorical, C, interval with equal variance, and D, ordinal with equal interval. Whenever we are talking about the chi-square test, that is used when you want to see if there is a relationship or difference between categories. I am repeating it. We are using chi-square in those conditions when we want to see there is any relationship or differences between categories. Specifically, when your data is in the form of counts or frequencies, like how many people or items fall into each categories, right? So when these kind of scenario, these kind of condition will be there, then we can use chi-square. But I can understand sometimes we are not able to relate something, so we need example. So we can one example to understand it, what I mean to say. Is there an association between gender, between male and female, and preferences for your type of snacks, chips and cookies? That means, what I mean to mention here, if you want to understand the relationship, and difference between gender and their preferences for the snacks, if you want to study this thing, this is categorical data, this is category, male, female, and then which kind of snacks like chips and cookies could be there. So whenever we are trying to understand the association between gender as well as type of snacks, this particular chi-square will be more suitable. That's why I told you chi-square test is used when you want to see if there is a relationship or difference between categories, right? So here you can see we are having these four options: A, continuous and normally distributed, B, nominal categorical, C, interval with equal variance, and D, ordinal with equal interval. And I am hopeful now you can answer them. So correct answer will be B, nominal categorical. B is the right answer. B is the right answer, nominal categorical. Next question is, which post-hoc test is most commonly used after significant ANOVA? You can focus on the question again. Which post-hoc test is most commonly used after significant ANOVA? Options are Bartlett test, option B, Chaff test, option C, Takey's HSD, and D, t-test. In comment box, you can mention or explanation of all these tests when we are using them. If you know it, you can mention where we can use and what is the meaning of it. But in this particular context, I would like to mention that when you run an ANOVA and find a significant result, then it tells you that at least one group is different, but it doesn't say which groups differ. This happens na? Whenever we run ANOVA, so you are identifying, right? If there are significant result, then you are identifying, and that tells you that at least one group is different, but that doesn't mentioning which group differ. That's where post-hoc test comes in. It compares each pair of group to see where the differences are. And the correct answer will be C. Right now I am mentioning what is the right answer, but I would like to request you to mention about all these tests in comment box. Now I am moving ahead. Okay, this will not be so difficult, right? We can answer them. Question is, when is a paired sample t-test appropriate? When is a paired sample t-test appropriate? The question is, when we will use paired sample t-test. Paired sample t-test. T-test again, t-test is a hypothesis testing. So when is a paired sample t-test appropriate? Your options are option A, comparing two unrelated samples, B, comparing more than two groups, C, measuring same objects at two time points, and option D, analyzing three or more categories. We have to understand what is paired t-test, and if you are aware about the paired t-test, that's great and you can answer this particular question. You will not face any difficulties. But if we don't know, then I always mentioning we are learning, right? So paired t-tests are used when the same subject are tested under two conditions. Again I said, sometime it's a limitations for us कि हम नहीं समझ पाते, right, just on the basis of their explanation. So we need example. So we can take one example. What I said first, paired t-test we are using when the same subject are tested under two conditions. For example, you measure a student's test score before and after a training session. So you conducted a test before training session, how much score they did, and after training session again you conducted a test and now how they are performing. In this case, paired t-test will be useful. Or we can say, suppose that you check blood pressure of patient before and after taking medication. Before taking medication, what is the blood pressure level, and after taking medication, they are going up, going down, or they are being stable. So paired t-tests are used, and we are using them when the same subject, right? Whenever, right, we are finding things or testing something in two conditions, right, of same subject. So paired t-tests are used when the same subject are tested under two different conditions. So correct answer, you can now find correct answer. First option mentioning comparing two unrelated samples. Option B mentioning comparing more than two groups. Option C mentioning measuring same subjects at two time points. And D, analyzing three or more categories. If you know the answer, I haven't mentioned the right answer, but now after explanation, you know the answer. That simply means sometime we are not able to answer just because we don't have, easiest understanding about something. If you know the answer, what is the right answer? If you know, C will be the right answer. That means if you will spend little bit of time on those components in which you are struggling and facing difficulties, you can manage them well, right? So finding or facing difficulties in something is the matter of, right, devoting time, or suppose that sometime if we are not identifying or understanding something by self, then, right, you have to contact your mentor. You are in college, you are in universities, you can talk to your mentor. They will explain you in easiest way. Like here you can see, now we all are aware, C will be the right answer. I would like to mention that every Thursday I am coming live on 3 o'clock, right, from last 3-4 weeks, and I'm having plan to maintain this, right. So if you need, and if our team will find that individual joining us and they are learning something, so we will extend this particular session. So you can join us on every Thursday at 3 o'clock, and we can discuss these things over there. Now next, 29th question is, if a person sets a conservative criterion in a signal detection task, which outcome is more likely? Question is, if a person sets a conservative criterion in a single detection task, which outcome is more likely? Option A is increased false alarm. Option B, increased hits. Option C, increased misses. Option D, no changes in direction. First of all, if you are aware, whenever we are using the signal detection concept, so there will be hit rate, there will be miss rate, there will be false alarm, and there will be correct rejection. Correct rejection. So these are the 4 conditions. Question is, if a person sets a conservative criterion in a signal detection task, which outcome is more likely? In signal detection task, you are trying to decide if a signal is present or not, right? If someone set a conservative criterion, that simply means they say yes only in those situations when they detect the sound, when they are very confident that yes, I heard the sound. So conservative criterion means what? Conservative criterion means individual will say yes only in those situations when that individual will be confident that yes, I heard the signal, I heard the sound. What it means? This means fewer false alarm, but also they will miss many signals, but they will not face false alarm, or fewer false alarm will be there. False alarm means what? False alarm means when signal is not present, individual saying yes, I heard something, I heard signal. So in this particular case, just because there is conservative criterion, so individual will say yes only in those situations when they are confident. So there will be lower false alarm, but at the same time they will also miss many signals. So correct answer you can see here. Now your options are increased false alarm, decreased hits, increased misses, no change in detection, and correct answer will be increased miss. What will be there? False alarm will reduce, fewer false alarm will be there, but we will miss many signals. We will miss many signals. That means increased misses. We will miss many signals. So C will be the right answer. Now the last question. Question is, in the adrenaline study, why did participants feel differently in different settings? Options are different doses. Option B, mood shift. Option C, contextual labeling. D, placebo use. Now there is a term adrenaline study. What it mean? Adrenaline we all are aware. But what is adrenaline study? So in a famous adrenaline study that is presented by Scatcher and Singer. So this is a study where people were given adrenaline without being told how it would affect them. That means individual or subject taken adrenaline, but researcher haven't told them that how this particular adrenaline will affect them. So after the shot, some were placed in a happy situation while others were placed in angry situation. And even though everyone had the same physical reaction, like after adrenaline, everyone's heartbeat went up, every individual's respiration went up, they reported different kind of feeling and different kind of emotions depending on where they was or what kind of situation, right, was there around them. What I mean to mention here, in adrenaline study, a Scatcher Singer given adrenaline dosage to group of people, half of individual placed in happy situation and half of individual placed in anger situation, and they don't know how adrenaline will react on their body, right? So each and every individual bodily changes was same. Heartbeat was higher and all the typical adrenaline related changes was there. But every individual presented different or reported different kind of emotions according to where they were and what kind of situation they encountered. So this happened just because they looked at what was happening around them, what happened in that particular situation, and that develops sense of their feeling, and this idea is called contextual labeling. What was the context? In which context individual adrenaline went up? So if there was high adrenaline in a happy moment, so there will be that kind of emotions. If any individual having high adrenaline, or if any individual taken adrenaline dose in angry situation, so they will realize or they will feel that kind of emotions. So we call them contextual labeling. What was the context over there? So now you can see different dosage, mood shift, contextual labeling, and D, placebo use, and correct answer is contextual labeling. So that's all for today, right? As I always mentioning, if you're having anything to say, anything to mention, you can put them in comment box, chat box. From last 3 weeks, every Thursday at 3 o'clock, we are conducting this particular session. So we are planning to continue this, right? And our team will decide it if students getting benefit from this session. So we are trying to continue this particular session. So you can join the next session at 3 o'clock next Thursday. And if you're having any doubt, any query, don't hesitate to mention in comment box and chat box, right? So thank you for joining this live session. Reshma mentioning good afternoon. Good afternoon, Reshma. Shaili, okay, Noha answering. Okay, so I hope you learned something new from this live session. So keep learning. As I mentioned in this particular session as well, whenever you are having opportunity to learn, right, get that opportunity. No matter you are doing mistakes or right, you are doing things correctly, don't hesitate to do mistakes, right? This is the great way to learn something new, experience something new. So don't hesitate to do mistakes. So that's all for today, right? See you in next session. Bye. Take care.